Skeletal muscle is under direct control of the somatic nervous system. The signal for a muscle fiber to contract originates in a motor neuron in the brain or spinal cord and is communicated to the fiber at its neuromuscular junction. Events at the neuromuscular junction lead to a muscle action potential. Excitation-contraction coupling describes the events that link this muscle action potential to activation of myofilaments within the muscle fiber, which causes a contraction. In order to understand excitation-contraction coupling, you must understand the anatomy of a muscle fiber. This image depicts a magnified view of the interior of a muscle fiber. The plasma membrane, called the sarcolemma, penetrates deep into the muscle fiber, forming T-tubules. The blue structure, which looks like a loose-knit sweater, is called the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium, which is released during a contraction at the bulging ends called the terminal cisternae. Together, the T-tubules and the terminal cisternae on either side form triads. Notice, however, that the majority of space inside the muscle fiber is occupied by myofibrils, which can shorten during contraction. Each myofibril is composed of a series of sarcomeres arranged end to end. A single sarcomere is made of thick filaments composed of myosin and thin filaments composed mostly of actin. Let's take a closer look at the thick and thin filaments. If we zoom in on a thick filament, we can see the myosin heads contain a binding site for actin. Similarly, a closer view of a thin filament shows actin molecules which contain an active site for myosin attachment. When myosin heads connect to the active site on actin, tension in the muscle fiber is generated, which is also known as a contraction. When the muscle fiber is in a relaxed state, the active sites are covered by a regulatory protein called tropomyosin, which is held in place by troponin. These regulatory proteins prevent the myosin heads from binding to the active sites on actin, thereby maintaining a relaxed state by preventing a contraction. Events at the neuromuscular junction set the stage for excitation-contraction coupling. When an action potential reaches the axon terminal, acetylcholine is released into the synaptic cleft, where it binds to receptors on the sarcolemma and triggers an action potential in the muscle fiber. Excitation-contraction coupling begins when the muscle action potential propagates along the sarcolemma and down the T-tubules. Let's zoom in on a triad to see what happens. When the action potential reaches the T-tubules, voltage-sensitive tubule proteins change shape, which opens calcium release channels in the terminal cisternae. These release channels allow calcium to flow into the cytosol of the muscle fiber. Let's take a closer look at the thick and thin filaments in the cytosol of the muscle fiber. Calcium in the cytosol binds to troponin, causing it to change shape and move tropomyosin away from the active sites on actin. The myosin heads now bind to the exposed active sites, forming a cross bridge between the thick and thin filaments. The myosin heads then use energy from ATP to pull on actin. This causes the thick and thin filaments to slide over one another, resulting in a contraction. Once nervous stimulation ceases, calcium release channels close. Calcium is continually pumped back into the sarcoplasmic reticulum by active transport. So with the release channels closed, cytosolic calcium returns to resting levels. If we take a closer look at the thick and thin filaments, we can see as the calcium concentration drops, calcium is released from troponin, causing a shape change that moves tropomyosin back over the active sites on actin. Once the active sites are covered, the myosin heads cannot form cross bridges, and the myofilaments can now slide back into their resting positions. You can see cross-bridge formation and muscle fiber contraction in this video. As long as calcium is present in the cytosol, the active sites are exposed, the myosin heads continue to form cross bridges, and the contraction continues. Once nervous stimulation stops and calcium is removed from the cytosol, the muscle relaxes. This process is called excitation-contraction coupling because it links the electrical signal to the muscle contraction. The excitation portion of this process is described as the action potential propagating along the sarcolemma and down the T-tubules. The contraction portion of this process is described as the myosin binding and cross-bridge formation. It is important to note that thick and thin filaments cannot respond directly to the electrical events associated with the action potential. Instead, contraction relies on the release of calcium. Therefore, calcium plays an extremely important role in this process by coupling excitation to the contraction. Let's summarize the steps in excitation-contraction coupling. Once the muscle action potential is generated near the neuromuscular junction, it propagates along the sarcolemma and down into T-tubules. This voltage change in the T-tubules causes calcium release channels in the terminal cisternae to open, releasing calcium into the cytosol of the muscle fiber. Calcium binds to troponin and causes it to change shape, removing the blocking action of tropomyosin. Now the active sites on thin filaments are exposed and the myosin heads can form cross bridges, resulting in a muscle fiber contraction. This process couples the excitation, or muscle action potential, to the muscle fiber contraction through the release of calcium.